Mechanism of hearing. Chapter 174. Introduction. Sound waves travel through external auditory meatus and produce vibrations in the tympanic membrane. Vibrations from tympanic membrane travel through malleus and incus and reach the stapes, resulting in the movement of stapes. Movements of stapes produce vibrations in the fluids of cochlea. These vibrations stimulate the hair cells in organ of Corti. This, in turn, causes generation of action potential, auditory impulses, in the auditory nerve fibers. When auditory impulses reach the cerebral cortex, the perception of hearing occurs. Thus, during the process of hearing, ear convert energy of sound waves into action potentials in auditory nerve fibers. This process is called sound transduction. Role of external ear. External ear directs the sound waves towards tympanic membrane. Sound waves produce pressure changes over the surface of tympanic membrane. Accumulation of wax prevents conduction of sound. In many animals, the auricle, pinna, can be turned to locate the source of sound. Auricle can be folded to avoid unwanted sound. But in man, the extrinsic and intrinsic muscles of auricle are rudimentary and the movement is not possible. Role of middle ear. Role of tympanic membrane. Due to the pressure changes produced by sound waves, tympanic membrane vibrates, i.e. it moves in and out of middle ear. Thus, tympanic membrane acts as a resonator that reproduces the vibration of sound. Role of auditory ossicles. Vibrations set up in tympanic membrane are transmitted through the malleus and incus and reach the stapes, causing to and fro movement of stapes against oval window and against perilymph present in scala vestibuli of cochlea. Impedance matching. Impedance matching is the process by which tympanic membrane and auditory ossicles convert the sound energy into mechanical vibrations in cochlear fluid with minimum loss of energy by matching the impedance offered by fluid. Impedance means obstruction or opposition to the passage of sound waves. When sound waves reach inner ear, the fluid, perilymph, in cochlea offers impedance, i.e. the fluid resists the transmission of sound due to its own inertia. Tympanic membrane and auditory ossicles effectively reduce the sound impedance. Sound waves are conducted from external ear to inner ear, with an impedance of only 40%. Remaining 60% of sound energy developed in tympanic membrane is transmitted to cochlear fluid by the ossicles. Thus, along with the help of tympanic membrane, ossicles match the impedance offered by fluid to a great extent. It is because the ossicles act like a lever system so that stapes exerts a greater force, pressure, against the cochlear fluid. This results in generation of vibrations in the cochlear fluid. The increased force is very essential to set up the vibrations in cochlear fluid because of higher inertia of the fluid. Force exerted by footplate of stapes on cochlear fluid is 17 to 22 times greater than the force exerted by sound waves at the tympanic membrane. It is because of two structural features of ossicles. Head of malleus is longer than long process of incus so that a higher force is generated in small structure. Surface area of tympanic membrane, 55 sqmm, is larger compared to that of footplate of stapes, 3.2 sqmm. So the pressure increases when force is applied to small area. Thus, the tympanic membrane and the auditory ossicles are capable of converting the sound energy into mechanical vibrations in cochlear fluid with minimum loss of energy. Significance of impedance matching. Impedance matching is the most important function of middle ear. Because of impedance matching, the sound waves, stimuli, are transmitted to cochlea with minimum loss of intensity. Without impedance matching, conductive deafness occurs. Types of conduction. Conduction of sound from external ear to internal ear through middle ear occurs by three routes: ossicular conduction, air conduction, bone conduction. Ossicular conduction. Ossicular conduction is the conduction of sound waves through middle ear by auditory ossicles. In normal conditions, the sound waves are conducted through auditory ossicles. Air conduction. Air conduction is the conduction of sound waves through air in middle ear. If the ossicular chain is broken, conduction occurs in an alternate route of air conduction. Air conduction is common in otosclerosis. Otosclerosis is the disease associated with fixation of stapes to oval window. Bone conduction. Bone conduction is the conduction of sound waves through middle ear by bones. It occurs when middle ear is affected. In this type of conduction, sound waves are transmitted to cochlear fluid by the vibrations set up in skull bones. Bone conduction is tested by placing vibrating tuning forks or other vibrating bodies directly on the skull. This route plays a role in transmission of extremely loud sounds. Role of eustachian tube. Eustachian tube is not concerned with hearing directly. However, it is responsible for equalizing the pressure on either side of tympanic membrane. Role of inner ear. Traveling wave. Movement of footplate of stapes against oval window causes movement of perilymph in scala vestibuli. This fluid does not move all the way from oval window to round window through helicotrema. It immediately hits the vestibular membrane near oval window. This causes movement of fluid in scala media, since the vestibular membrane is flexible. Movement of fluid in scala media causes bulging of basal portion of basilar membrane towards scala tympani, Fig 174.1. Bulging of basilar membrane increases the elastic tension in basilar fibers in that portion of basilar membrane. Elastic tension in basilar fibers initiates a wave, which travels along basilar membrane towards the helicotrema like that of arterial pulse wave. It is called traveling wave. Resonance point. Resonance point is the part of basilar membrane, which is activated by traveling wave. In the beginning, each traveling wave is weak, Fig 174.2. While traveling through basilar membrane from base towards apex, helicotrema, the wave becomes stronger and stronger, and at one point, resonance point, of basilar membrane, it becomes very strong and activates the basilar membrane. This resonance point of basilar membrane immediately vibrates back and forth. The traveling wave stops here and does not travel further. Distance between stapes and resonance point is inversely proportional to frequency of sound waves reaching the ear. Traveling wave generated by high-pitched sound disappears near the base of the cochlea. Wave generated by medium-pitched sound reaches half of the way, and the wave generated by low-pitched sound travels the entire distance of basilar membrane. Excitation of hair cells. Stereocilia of hair cells in organ of Corti are embedded in tectorial membrane. Hair cells are tightly fixed by cuticular lamina reticularis and the pillar cells or rods of Corti. When traveling wave causes vibration of basilar membrane at the resonance point, the basilar fiber, rods of Corti, hair cells, and lamina reticularis move as a single unit. It causes movements of stereocilia leading to excitement of hair cells and generation of receptor potential. Electrical events during process of hearing. Sound transduction. Sound transduction is a type of sensory transduction, Chapter 139, in the hair cell, receptor cells, in organ of Corti, by which the sound energy is converted into action potentials in the auditory nerve fiber. Electrical events of sound transduction. Three types of electrical events that occur during sound transduction are, receptor potential or the cochlear microphonic potential, endocochlear potential or endolymphatic potential, action potential in auditory nerve fiber, receptor potential or cochlear microphonic potential. Receptor potential or cochlear microphonic potential is the mild depolarization that is developed in the hair cells of cochlea when sound waves are transmitted to internal ear. Resting membrane potential in hair cells is 60 MV. Sensory transduction mechanism in cochlear receptor cells is different from the mechanism in other sensory receptors. When sound waves reach internal ear, traveling wave is produced. It causes vibration of basilar membrane, which moves stereocilia of hair cells away from modiolus, towards kinocilium. It causes opening of mechanical ligated potassium channels, Chapter 3, and influx of potassium ions from endolymph, which contains large amount of potassium ions. Influx of potassium ions causes development of mild depolarization, receptor potential, in hair cells up to 50 MV. Cochlear microphonic potential is non-propagative. But, it causes generation of action potential in auditory nerve fiber. Due to depolarization, hair cells release a neurotransmitter, which generates action potential in the auditory nerve fibers. Probable neurotransmitter may be glutamate. Movement of stereocilia away from modiolus, towards kinocilium, causes depolarization in hair cells. Movement of stereocilia in the opposite direction, away from kinocilium, causes hyperpolarization. Ionic basis of hyperpolarization is not clearly known. It is suggested that calcium plays an important role in this process. Hyperpolarization in hair cells stops generation of action potential in auditory nerve fiber. Role of hair cells. Inner hair cells and outer hair cells have different roles during sound transduction. Role of inner hair cells. Inner hair cells are responsible for sound transduction, i.e. these receptor cells are the primary sensory cells, which cause the generation of action potential in auditory nerve fibers. Role of outer hair cells. Outer hair cells have a different action. These hair cells are shortened during depolarization and elongated during hyperpolarization. This process is called electromotility or mechanoelectrical transduction. This action of outer hair cells facilitates the movement of basilar membrane and increases the amplitude and sharpness of sound. Hence, the outer hair cells are collectively called cochlear amplifier. The electromotility of hair cell is due to the presence of a contractile protein, prestin, named after a musical notation presto. Role of efferent nerve fibers of hair cells. Efferent nerve fibers, chapter 173, of hair cells also play important role during sound transduction by releasing acetylcholine. Efferent nerve fiber to inner hair cell terminates on the auditory, afferent, nerve fiber where it leaves the inner hair cell. It controls the generation of action potential in auditory nerve fiber by inhibiting the release of glutamate from inner hair cells. Efferent nerve fiber to outer hair cell terminates directly on the cell body. It inhibits the electromotility of this cell. Endocochlear potential or endolymphatic potential. Endocochlear or endolymphatic potential is the electrical potential developed in fluids outside the hair cells. Cochlear fluids. Cochlear fluids are the extracellular fluids in the inner ear. These fluids are perilymph and endolymph, which have different composition. Perilymph. Scala vestibuli and scala tympani are filled with perilymph, which is similar to ECF in composition with high concentration of sodium ions. Endolymph. Scala media is filled with endolymph, which contains high concentration of potassium and low concentration of sodium. It is due to continuous secretion of potassium ions by stria vascularis into scala media. Electrical potential. Difference in potassium concentration is responsible for the development of an electrical potential difference between endolymph and perilymph. Potential in endolymph is positive up to plus 80 MV. Significance of endocochlear potential. Lower portion of the hair cells is bathed by perilymph. Head portion of hair cells penetrates the lamina reticularis and it is bathed by endolymph, Fig 172.3. Endolymph has a positive potential, plus 80 MV. So inside the hair cells, the electrical potential is 60 MV in comparison to that of perilymph and 140 MV in comparison to that of endolymph. High potential difference sensitizes the hair cells so that, the excitability of hair cells increases. It also increases the response of cells even to slight movement of stereocilia. Action potential in auditory nerve fiber. Action potential in auditory nerve fiber is generated by cochlear microphonic potential. It obeys all or none law and has a definite threshold and refractory period. Action potential to a click sound with moderate intensity level consists of three successive spike potentials called N1, N2, N3 representing synchronous repetitive firing in many fibers. At high frequency, the synchronization of action potential disappears and single spike occurs. Action potential appears 0.5 to 1 millisecond after the development of cochlear microphonic potential. Properties of sound. Sound has two basic properties: pitch, which depends upon the frequency of sound waves. Frequency of sound is expressed in hertz. Frequency of sound audible to human ear lies between 20 and 20,000 Hz or cycles/second. The range of greatest sensitivity lies between 2000 and 3000 Hz, cycles/second. Loudness or intensity, which depends upon the amplitude of sound waves. It is expressed in decibel, dB. The threshold intensity of sound wave is not constant. It varies in accordance to the frequency of sound. Appreciation of pitch of the sound, theories of hearing. Many theories are postulated to explain the mechanism by which the pitch of the sound is appreciated or the frequency is analyzed. These theories are generally classified into two groups. According to the first group, the analysis of sound frequency is the function of cerebral cortex and the cochlea merely transmits the sound. According to the second group of theories, the frequency analysis is done by cochlea, which later sends the information to cerebral cortex. Theories of first group. Telephone theory of Rutherford. Telephone theory was postulated by Rutherford in 1880. It is also called frequency theory. According to this theory, the cochlea plays a simple role of a telephone transmitter. In telephone, sound vibrations are converted into electrical impulses, which are transmitted by cables to the receiving end. There the receiver instrument converts the electrical impulses back into sound waves. Similarly, cochlea just converts the sound waves into electrical impulses of same frequency. Impulses are transmitted by auditory nerve fibers to cerebral cortex, where perception and analysis of sound occur. It is believed that the nerve fibers can transmit maximum of 1000 impulses per second. Thus, the telephone theory fails to explain the transmission of sound waves with frequency above 1000 cycles per second. So, a second theory was postulated. Volley theory. In 1949, Weaver postulated this theory. Volley means groups. According to this theory, the impulses of sound waves with frequency above 1000 cycles per second are transmitted by different groups of nerve fibers. However, this theory has no evidence to prove it. Thus, these two theories were not accepted by many physiologists. Theories of second group. Resonance theory of Helmholtz. Resonance theory was the first theory of hearing to emerge in 1863. According to Helmholtz, analysis of sound frequency is the function of cochlea. Basilar membrane contains many basilar fibers. Helmholtz named these basilar fibers resonators and compared them with the resonators of piano. When a string in piano is struck, sound with a particular note is produced. Similarly, when the sound with a particular frequency is applied, the basilar fibers in a particular portion of basilar membrane are stimulated. Resonance theory was not accepted because the individual resonators could not be identified in cochlea. Gradually, this theory was modified into another theory called the place theory, which is more widely accepted. Place theory. According to this theory, nerve fibers from different portions, places, of organ of Corti on basilar membrane give response to sounds of different frequency. Accordingly, corresponding nerve fiber from organ of Corti gives information to the brain regarding the portion of organ of Corti that is stimulated. Many experimental evidences are available to support place theory. Experimental evidences supporting place theory. If a person is exposed to a loud noise of a particular frequency for a long period, he becomes deaf for that frequency. It is found that the specific portion of organ of Corti is destroyed, as in the case of Boilermaker's disease. In experimental animals, destruction of a portion of organ of Corti occurs by exposing the animal to loud noise of a particular frequency. In human high-tone deafness, there is degeneration of organ of Corti near the base of cochlea or degeneration of nerve supplying the cochlea near the base during exposure to high-frequency sound. Cochlear microphonic potentials show greater voltage in hair cells near base of the cochlea. Also, During the exposure to low-frequency sound, cochlear microphonic potentials show greater voltage in hair cells near apex of the cochlea. There is point-to-point representation of basilar membrane in auditory cortex. Traveling wave theory. From place theory, emerged yet another theory called the traveling wave theory. This theory explains how the traveling wave is generated in the basilar membrane. Development, generation, movement, and disappearance of traveling wave are already described earlier in this chapter. Appreciation of loudness of sound. Appreciation of loudness of sound depends upon the activities of auditory nerve fibers. Intensity or loudness of sound correlates with two factors: 1. Rate of discharge from the individual fibers of auditory nerve. Total number of nerve fibers discharging. When loudness of sound increases, it produces large vibrations, which spread over longer area of basilar membrane. This activates large number of hair cells and recruits more number of auditory nerve fibers. So, the frequency of action potential is also increased. Localization of sound. Sound localization is the ability to detect the source from where sound is produced or the direction through which sound wave is traveling. It is important for survival and it helps to protect us from moving objects such as vehicles. Cerebral cortex and medial geniculate body are responsible for localization of sound.